What if AI could design gene therapies so precise they eliminate chronic pain while leaving other senses completely intact? And what if the secret to treating Parkinson's disease lies hidden in the genome of a mouse? Hello, you're listening to where What If becomes What's Next from Carnegie Mellon University, where we ask the bold questions that become innovations for the betterment of humanity. I'm your host, Randy Scott, and today we're exploring new advances in healthcare and how AI is revolutionizing genetic medicine through incredibly precise targeting and cross-species genomic research. Our guest is Andreas Pfenning, an associate professor in the Computational Biology Department and School of Computer Science at Carnegie Mellon University, where Professor Pfenning also runs an experimental lab that is developing new AI-powered methods for disease detection and treatment. Andreas, what areas of healthcare are you working on in your lab?
One of those areas is gene therapies. Gene therapies have had a lot of promise, but there's also a lot of barriers to gene therapies being used every day for a lot of the health conditions people really care about. And one of those barriers that I think AI and integrating AI with experimentation can really help with is in making gene therapies more targeted. When I'm talking about gene therapies, you're talking about basically taking cells that might be somehow not doing their job within the body, and you're trying to put them in the right state or getting them to do their job by delivering a gene to them. And sometimes that might be a gene that these cells are missing because of the genetics of the subject. Or it could be basically trying to introduce a new gene that maybe, for example, in the case of neurological or psychiatric disorders, makes a type of neuron more active that allows it to do its function, or maybe shut down a neuron that isn't doing what it should.
When cells malfunction because of a missing or faulty gene, what happens to the person? Is that what disease looks like at the cellular level?
It really depends on the disease and the condition. It's quite broad, but it could manifest as cancer. It could manifest as a rare disorder. But, a lot of basically having too much or too little of a gene can also have, really big effects for common disorders. So it's really one of the, let's say, major ways in which diseases come about within the human population.
Got it. So what are you working on to make gene therapies more effective in fighting diseases?
As I mentioned, one of the barriers is to, let's say, more effective or broader sets of gene therapies is the targeting. So just at a high level, let's say you deliver a gene to a cell that the gene is missing in, right? Then maybe that cell starts to work better and you can cure or effectively treat the disease, and that's the desired outcome. On the other hand, it could be that you deliver that gene to another cell where that gene isn't supposed to be, and you can get toxic side effects. And those toxic side effects are still, a major barrier to a lot of gene therapies being more broadly adopted. One of the things that my lab does is basically using AI or artificial intelligence to design gene therapies that make them more targeted and more specific.
Okay, but what does that involve? I expect it's both identifying which cells need the therapy and developing better ways to deliver it to those specific cells, right?
That's absolutely right, and it's all of the above. A lot of different labs, my lab included, we do work on trying to identify exactly which cells need the gene and exactly which cells are causing the disease state. Now, where my lab focuses is actually not in the delivery, but the activation. Essentially, everybody has their own genome, and that genome is going to be mostly the same across all of the different types of cells in the body. But what makes, let's say, a neuron different from a skin cell, different from any other type of cell, is that basically there are different sets of proteins or genes active in these different cells that help them to perform their function. And so all of these different functions are essentially encoded in the genome, and we like to think of it as a language that's encoded in the genome. So what we do is basically we collect data from different types of cells in different parts of the brain especially, or different immune cells under different conditions. And then we learn the language of that cell to try to better understand what's going on. And once we learn the language of that cell in the genome, we can then use that to basically design more efficient gene therapies.
Okay, Andreas, how does AI help with that?
If you deliver your gene therapy to a whole group of cells, we can include a little signature designed by machine learning that basically says we're only going to activate the gene in the cells that need it, but we're not going to activate the gene in the other cells.
And that reduces the risk of toxic side effects and make the treatment more successful. Is this approach currently being used for any specific diseases?
So I think one of the cases where we're really trying to, let's say, go for all the way from the initial idea to a downstream therapy is for treatment of chronic pain. And a lot of chronic pain is controlled by the spinal cord. And the reason why the spinal cord, let's say, is a good test case for some of this technology is how complicated and diverse the spinal cord is. So in the spinal cord, you have some cells that transmit signals for chronic pain, which of course you want to inhibit. But then there are other cells that are involved in normal touch or sensation, sensing hot and cold. Other cells involved in acute pain, which you actually don't want to modulate or mess with because that can be important information that our bodies need to, stop touching a hot stove. The spinal cord also, of course, mediates movement, and you don't want to inhibit things like breathing when you're delivering a treatment.
So given how sensitive and dangerous it is to intervene in the spine, what does the actual delivery of this genetic therapy look like? Are we talking about big needles or a surgical procedure?
Well, so first I want to mention that we're still building up towards a specific therapeutic, but ultimately what this would look like is delivering something microscopic that's in a big needle. So you were right on both points.
Well, pain affects millions of people, giving you lots of data to work with. But with rare diseases, you have rare sample sizes. I understand that CMU has developed with other partner research institutions a new AI-powered tool called KGWAS that helps identify genetic problems even with limited sample sizes. KGWAS stands for Knowledge Graph Genome-Wide Association Study. Can you tell us about that and how it will be used?
I think the spinal cord is actually a good example of that too. So one of the first steps in starting this line of work in the spinal cord is people actually didn't have a map of the type of cells that existed in the spinal cord. I just want to mention that this work is being done in collaboration with Becky Seal's group at the University of Pittsburgh. So after we had that map of different cell types, we basically needed to know, okay, among these cell types, which ones are the ones that are likely transmitting pain signals? And that's where work like KGWAS can come in really handy. KGWAS and similar methods can relate that map to signals coming from genome-wide association studies. So what those studies do is they basically take subjects with and without a particular disease or disorder, and then they run association tests or statistical tests across the entire genome. And then you can ultimately get parts of the genome that are related to the disease or are not related to the disease.
Would KGWAS have been possible without machine learning and AI?
I would say that for a lot of the areas that we work on, you can, let's say, get an initial prediction or some kind of prediction without machine learning. But in the example of KGWAS and some of the other methods that my lab has developed and that we use, machine learning gets us much, much closer to a correct answer. And if you're dealing with something like modulating cells or inhibiting cells in the spinal cord, you really want to be sure that the cells you're going after are the right ones and not the wrong ones.
Well, that sounds like it could also be helpful in how we identify and treat rare diseases. Is that right?
It does make possible interpreting genetic signals for more rare diseases or cases where the information is limited, and that can help to identify the right cell types, the right genes, and to link everything together.
It sounds like KGWAS and AI can help scientists do more with less.
I would say one of the ways in which I think it can be broadly useful is in basically filling in the gaps and making predictions when, let's say, some of the more advanced technology isn't available, right? And in that way, I think it can democratize a lot of medical knowledge. You don't have the availability of a big fancy scanning machine. to tell you everything about your brain and its state and how that might relate to a potential neurological or psychiatric disorder, right? I think it's now possible to take less advanced technology and combine that with machine learning to basically infer what the fancy machine would have told you. So I do think there's a lot of promise broadly in healthcare in terms of using AI to augment existing technologies and making diagnoses and information more accessible.
Well, does this also mean we can now pursue genetic research on rare diseases that wasn't feasible before due to data or resource limitations?
I think that's absolutely fair. There are some diseases that we just have much more limited genetic information for, and KGWAS can basically supplement that by drawing from other databases and other studies to give you a picture of which cell types and which genes are actually most likely to be involved in the disease. And that can provide a really important foundation for starting to treat them, like using some of the gene therapy techniques that my lab and others use to target the specific cell types, or using other things like small molecules or drugs to target some of the genes that KGWAS can implicate.
This seems like a good place to take a quick break. And when we come back, we'll talk with Professor Pfenning about how mapping the genomes of more than 500 vertebrate species could unlock genetic secrets that could transform human medicine.
This podcast is brought to you by Carnegie Mellon University. Located in Pittsburgh, Pennsylvania, CMU is a private, international research university, consistently ranked among the top universities in the world. Its trailblazing research is focused on creating, defining, and accelerating the future of artificial intelligence. For more information, please visit ai.cmu.edu.
Welcome back. You're listening to Where What If Becomes What's Next from Carnegie Mellon University. We're talking with Andreas Pfenning, an associate professor in the Computational Biology Department and School of Computer Science at CMU, about new AI-powered technologies in genetic mapping and gene therapies. Let's switch gears, or should I say, let's switch species, and talk about your fascinating work with mapping the genetics of different animal species.
My laboratory is heavily involved in a large international project called the Vertebrate Genome Project. Now, the broader goal is to basically get a high-quality genome sequence for all vertebrate species on Earth, right? So we're talking about mammals like humans, birds, reptiles, amphibians, fish, and provide the foundation for conducting really important science experiments and answering really fundamental questions in biology. So, the human genome was initially sequenced and finished back in 2001. And if you study human genetics, you know just how crucial it is for almost everything that we do in that field.
What's the impact of mapping all these vertebrate species? Will this lead to discoveries that affect human medicine?
I would say absolutely. So one of the things that we're finding relates to some of the gene therapy techniques that I was talking to you about before. We had a really surprising finding a few years ago from that. And the surprising finding is that even if the individual nucleotides of the genome, even if those are very different between human or mice, or human and elephant, or human and black rhino, let's say, even if those individual nucleotides are very different, we found that the language of a particular region can be the same or constant. So in other words, one part of the genome that might be active in a particular cell type of the human brain, looking at that same part of the genome in the mouse, that part of the genome can also be very likely to be active in the same brain region and same cell type in mouse. In the case of the spinal cord research, for example, we initially are testing the technology in mice. And we've been able to use machine learning to target the specific cell type that we predict to be involved in chronic pain. And as we expect, the chronic pain in the mice is blocked, but the movement and normal touch or sensation and other things like itch are actually preserved. So I view that as a really important milestone because this is a technology that's readily adaptable to human.
That sounds incredibly promising for chronic pain. Is there also a broader vision of what can be done from this genetic research?
What we're hoping to do more broadly, and a lot of this is work with the NIH Brain Initiative and Cell Armamentarium Consortium, is actually build an entire toolkit that can serve as the foundation for treating a number of neurological and psychiatric disorders. So much of the work has been focused on targeting gene X or gene Y, and that's of course really important. But we feel that if we can develop more targeted therapeutics that go after the specific cell types that are causing the disease, then we can open up avenues for a lot of neurological and psychiatric disorders. So we have projects in parallel going on for Parkinson's disease, projects in parallel going on for Alzheimer's disease, where again, we're trying to target the cells that we think are driving the disease process forward and leaving the other cells that are healthy to perform their normal functions.
The Human Genome Project took 13 years to complete, and that was just for one species. The Vertebrate Genomes Project is mapping hundreds, if not thousands, of species. It sounds daunting.
This is just an enormous effort, and I think one of the encouraging things is that it really did take a ton of international cooperation. The Vertebrate Genome Project has been ramping up since around 2015, 2016, and there's a ton that goes into getting a resource like this. So first of all, you need the samples from the different species. And that, of course, has to be an international effort. You have people going into a rainforest to collect samples and other people trekking into the desert. And of course, other people in zoos or maybe even in backyards getting samples as well. So you need the raw material to get the genome. There were also technical challenges that the consortium needed to solve on doing the sequencing itself. So how do you do that at scale? Given the fact that the human genome took so long, how can you do it right the first time with advanced genome sequencing technologies? And then once you had the raw material, the genome sequences, you then had to kind of assemble them into full genomes. And that was also a huge effort that took a lot of algorithmic development, a lot of computational power to get those full genomes.
I imagine AI and machine learning play a huge role in making this work possible, right?
Yeah, absolutely. And again, that's one of the things that my lab specializes in. So one example is it can be both practically and ethically unavailable to do experiments directly in black rhino tissue. But you might want to learn about black rhino biology. So for example, my lab has developed methods that take information from, for example, the human genome and the mouse genome, and then use that to build models that can make predictions about what parts of the black rhino genome are going to be active and infer aspects of their biology.
When do you expect this massive project to be completed?
That's a good question. So we are working very hard on trying to put together, what we're calling the first phase of the project. So basically publishing very high quality genomes from 580 different vertebrate species and integrating that with existing tools. Not willing to provide a specific date, but I would say that we're actively working on the genomes and the paper, and we kind of hope to have that out within the next half year or year.
So once you've mapped 500 plus species, you'll be able to see intersections among all these genomes. Could this lead to breakthroughs in treatment of human diseases, for example, arthritis, heart disease, neurological disorders, by mapping their genetic roots?
We're conducting research right now on longevity, and we're finding that some of the parts of the genome that relate to whether species live longer or shorter, that those can be related to things like Alzheimer's disease and Parkinson's disease. So I really think that there are many different parallels between the function of genomes and what we can learn from the enormous diversity of vertebrate species. And we can use that to directly tell us what some of the mutations are doing in the human genome and how those might relate to different disease processes.
Looking toward the future, what emerging technologies or approaches in genetic therapy excite you the most?
A lot of what we're hearing about these days is in AI and advances in AI. And I think those are extremely important and also something my lab focuses on. An area that I think is actually often, let's say, underhyped is some of the experimental technologies. For example, we have been working on the AI technology I talked about to make gene therapies more targeted. But my group and other groups were really laboriously testing, each of these sequences one at a time to see how targeted they were. So one technology my lab has been working on is basically finding ways to test the function of dozens or hundreds of sequences at once, so that we're not just getting information one at a time, but the experimental technology scales with the artificial intelligence. And that's actually where I think a lot of the future is, not just building bigger and better artificial intelligence models, even though, again, I think that's really important, but a more tight integration of the AI models with new technologies that allow you to collect larger data sets, and also specifically data sets that tell you a lot of information about the disease or biological system you're trying to study.
What's the pathway for bringing these AI-powered tools to helping patients? Do these tools go to clinical researchers, pharmaceutical companies, or directly to doctors?
So the first stage is that we're making these tools available to researchers doing studies in mice, in non-human primates, to basically allow them to work at the biology of the system. The next step is to make them available to pharmaceutical companies that will kind of allow them to make their therapeutics less toxic and more targeted. And then after that development, then they can go into the clinic where they're actually in the hands of physicians and helping patients.
Okay, let's jump ahead 10 or 15 years. Are there certain diseases that could actually be cured or at least more effectively treated because of your lab's work?
I would say in the case of Parkinson's disease specifically, so we collaborated using a different technology, but also a technology to target specific cell types with the laboratory of Erin Gittes. So she's in biology and does neuroscience work here at Carnegie Mellon. And in working with her, we basically developed a targeted stimulation technique that was able to reverse a lot of the symptoms of Parkinson's disease in mice. And now there's a clinical trial going on in the human population that's also looking to see if that will work for humans. But the initial data and results are actually quite promising that the new type of stimulation is safe and effective. So I would say that's already a great proof of concept and something that's in the clinic right now, where AI and the ability to target specific types of cells is already having an impact on patients' lives and is being more rigorously tested in the right way.
Well, that's exciting. Well, that concludes our episode. Thanks to our guest, Professor Andreas Pfenning, and thank you to you for listening. Please check the show notes where you'll find links to additional resources for our episode. And to learn more or reach out to us with a story idea or comments, just visit cmu.edu/whatsnextpodcast. If you're enjoying the show, please rate and review us on your favorite podcast site, and be sure to share with your friends. And be sure to subscribe too, so you'll never miss an episode. I'm Randy Scott, and you've been listening to Where What If Becomes What's Next from Carnegie Mellon University, where we are asking the bold questions that become innovations for the betterment of humanity. Stay tuned and stay curious.